and I'd like to thank Gymnasia, the Foundation, to make this event possible. And just, I mean, you think about it. English is not your first language. English is not my first language. So it really doesn't matter if you or I make any kind of mistakes in me presenting or you asking. Okay? So don't be shy. Whenever you have a question, just ask and I'll try to answer as good as I can. We want to talk about Alzheimer's disease this week, and I don't know whether you have heard of, well, you've certainly heard of Alzheimer's disease, but I don't know whether you have any direct contact with patients or whether you know how Alzheimer's disease really develops. One thing you know for certain is that Alzheimer's disease develops in the brain, and only in the brain. Now, thinking of the brain is hard. Now, you could go to any anatomy class and they will tell you, okay, this is the frontal cortex, this is the hippocampus, whatever. They can pinpoint it to you. Then you go to a physiologist and he will tell you, okay, you need an action potential to drive any kind of information along an axon. Fine. Then you go to a biochemist and they will tell you, you need certain receptors to move information into a cell. That is all true. But can anyone tell me how we think? I have no clue. I mean, why are we generating information? How are we storing information? We have no clue how that works. So we are dealing here with a disease in an organ that we do not understand. The brain is the most complex structure in the whole universe. That's what we say. Meaning, we don't know anything about it. We know bits and pieces, but we don't know how it really works altogether. And that is a problem. I mean, thinking of it, dealing with a disease we cannot cure in an organ we do not understand. I mean, that's really the worst scenario you can ask for. But anyway, we will try to understand the disease as I see it. That doesn't mean that my view is correct, but I'll try to explain to you how most of the researchers see Alzheimer's disease, and we can even discuss some aspects that other people favor, maybe. Okay, now thinking of the brain. What is the brain? If you think how the brain looks like or how it should work, here it is. It is, in general, the best protected organ in our body. It is protected by the skull and by a fluid. No other organ is protected as well as the brain. So then in general, if you touch a brain, it feels like a ripe avocado. Pretty much the same texture as an avocado. When you think of the brain mass, it is only 3% of your body, but it consumes 40% of your energy all the time. Meaning this 40% energy that you're consuming, you could run a 40-watt light bulb on it constantly. This much energy is used up by your brain when you are actually not using it. So now your light bulb should burn much lighter than 40 watts, so maybe 60 watts, I don't know. And of course it looks like a very huge walnut. And that is pretty much a good description of the brain. Okay, we know a little bit more, but not much more, to be honest. We know it's around 1 kilogram in weight, and we can slice it up and look at structures, but that's pretty much it. But we can do here a little quiz, if you like. Can anyone tell me how many neurons we have in one brain? So just feel free to say any number. I mean,
few minutes.
Not bad. 10 to the 11th, meaning 100 billion neurons in one brain. Do we have more neurons or more glia cells in the brain? More glia cells, right. Any clue how many more? How much more? 10 times more, right. So we have 10 times more glia cells in the brain But nobody cares about glial cells. Right? When we talk about the brain, we always talk about neurons. Nobody talks about glial cells. Why? I don't know. But we completely forget about it. Okay, so we have in one brain 100 billion neurons. Now you know neurons have axons, dendrites, and all these protrusions. So when you put all of them from one brain in one line, how far would it reach? Would it be from here to Moscow, from here to Berlin, from here to New York, from here to around the world?
3 times around the world.
3 times around the world, meaning 120,000 kilometers? Okay? 500,000 kilometers. So that means from here to the moon and half the way back. From one brain. How many synapses do we have in the brain? So synapses are the contact points between neurons. How many of them? Do we have the same number as neurons? More or less? Much more, correct. So 10 to the 14th. So that's a number I have a problem with. So if you would tell me I have 10 to the 14th dollars, I know I'm rich. But I don't know how rich I am. Okay? So think about it. If you would count one synapse per second in one brain, for how long would you count? Okay, 10 to the 14 seconds, I know. But how long is that? Is that a day? Is that a week? Is that a month? Is that a year? 1,000 years? How long?
Few lives.
3 nights?
Few lives.
Oh, few lifetimes, okay. Few lifetimes could be 150 years? Kind of. 3 million years. Okay, so that is the amount of synapses you have if you would count one synapse per second in one brain. Do you know what the size is of a synapse? If you think about a cell, a regular cell is about 10 micrometers maybe. Some are bigger, some are smaller. A synapse is 300 nanometers. And that is a problem in the case if you look through a light microscope. Because this is the wavelength of visual light, meaning you cannot see a synapse with a regular light microscope. You need a higher magnification, okay, due to the wavelength restriction. And if a synapse would be 1 centimeter instead of 0.3 micrometers, your brain would be 4 kilometers long. So these numbers I just put up there to show you how complex that organ is. And we're only focusing on neurons, right? No glia cells. So that is a problem. But anyway, we all try to understand the brain, and we even want to generate a computer that works as a brain, right? And I'm pretty convinced that at least 60% of you are sure that we will generate a computer in the near future that works like a brain. No way. I don't mind. If you build that thing, fine. No problem, okay? I just don't believe in it. But anyway, if you show me it works, fine. I'm convinced, but not before that. So, if you think about a computer and a brain, a computer is actually a pretty dumb thing. The only thing that a computer can do is 1 and 0. Nothing else, right? It's a digital system. So you can switch it on, you can switch it off. So that is digital system. And a brain or a neuron can actually do the same. If you think of, if my arm here and my hand, this is a neuron, this is a soma, these are the dendrites, and my arm is the axon. Then what happens in a neuron regularly is that you have an action potential that is generated at the soma and then run through the axon to the end of the axon. That is a digital signal, right? You turn it on, action potential, you turn it off. That is a computer. The problem is, at the very beginning, the dendrites, you have millions of different information coming into one neuron through the dendrites. And for whatever reason, in the soma, something decides which signal to use. We have no clue how that switch works. And that is something I presume a computer can't do right away. Anyway, a computer can tell me in the next picture what is wrong. And please just yell into the room what you think is wrong on the next picture. Don't fear anything, just tell me right away. Don't think long about it. What's wrong about that? Upside down, right. See that? So, you were all convinced that this is Abraham Lincoln, only upside down. But the eyebrows here were on the wrong side there as well. Who saw that only after I pointed that out? Honestly. Yeah, good. So, what you are no computers, meaning what you have is imagination, right? You know how a picture should look like, and then you just take your visual image and put it on top of it. So when you, for example, look into some newspapers, sometimes they have these quizzes there. 2 pictures on the left-hand side and 1 on the right-hand side. And then they say on the right-hand side there are 10 things missing in that picture, right? What you do is you look at first on the left-hand side, right-hand side, everything there. Yes, because you got the visual in your head already. Therefore, you cannot see the difference. No computer would ever be able to do that. Okay, with some software and modifications whatsoever, right? But at the moment, that is what your brain is capable of doing. So we can really imagine stuff. And computers are pretty dumb. Well, in theory. So, okay. I hope I could convince you that the brain is complex and we don't know that much about it. We now want to focus on the disease, and it is named after Alois Alzheimer. You see everything written on here is in German, and it has a reason that it's in German, because he was a German doctor, and he was working in Frankfurt at the time when his first patient was brought to the facility. And it was Auguste Deter, and she was, at those days, people would say she was weird, right? And at those days, everywhere in the world, when people were weird, meant they were basically housed somewhere. It's pretty much a jail where they were put in. So there was no treatment whatsoever, but if you do not belong or do not behave in a proper way in society, doesn't matter whether you were a criminal or sick. And if you couldn't treat these sickness, you were jailed. And that was pretty much it what happened with all these diseases. And that is one reason that we know so little about these diseases. I mean, this was a common concept, at least in Germany, until 60, 70 years ago. And you think about it. I mean, it's terrible if this happens to a person. And Alzheimer was a peculiar doctor because he was really interested in what was going on with Auguste Deter. She was deteriorating very, very fast. So her brain, her mind was going down in a rate he hasn't seen before. So he was just making marks and trying to talk to her, and he wrote everything down that she mentioned, and it was really, there was, after a few years, really nothing left she could communicate. And that was interesting to him. And he presented then this finding on a conference. And what he presented there were these original drawings he made. So nowadays, when you look into a microscope and you see a cell, you take a picture with a whatever camera, right? Those days, he had no camera. He had a microscope, and he had to draw everything. I remember when I was studying, my teachers told me, you have to draw every little detail. I hated it, right? I hated it. 5 years ago, I went through my study and threw out all the old stuff. Actually, I wanted to throw it out. And then I got back to my old literature that I was drawing myself. It was so beautiful. I couldn't even imagine that I could have done that. But it was really, really cool. And the coolness about it is, when you draw something, you pay attention. And if you think about it, you sit on a microscope and you take a picture. You see, okay, cell, click. Then you go in, cell, click, right? You really don't care what's under there. But here, he had to draw everything by hand, and he was seeing things like this here. This is clearly a neuron, and he painted some dark stuff in that neuron. And that is up to now one histopathological hallmark of an Alzheimer's diseased person. You have to have neurons with these dark stuff in those neurons. And you have to have these things here. These are black, really black, dark dots in the brain of an Alzheimer's disease person. That is another histopathological hallmark. This is an Alzheimer plaque. So Alzheimer was seeing something like that when he was looking after Auguste Deter's death into the brain of Auguste Deter. He saw these dark plaques, and he saw these dark stripes here, which are tangles. And what that means to the disease, I will explain to you in a minute. Up here you see two brains, and a lot of people think that this brain is a typical Alzheimer's disease brain. It is not. It is a typical brain of a very old person. The older you get, the more brain mass you're going to lose. So when you're 90 and 95, you have lost 20 to 30% of your original brain mass. You can't do anything about it. Right? So this is not typical Alzheimer's disease, it's just old. This is a young brain. That's the difference. But in an Alzheimer's brain, you always have to find this. Otherwise, it's not an Alzheimer's disease patient. And this is what Alois Alzheimer described in the very beginning. And he described that in his first publication, and it's a publication in Germany at those days, and the name is Über eine eigenartige Erkrankung der Hirnrinde, meaning it's a weird disease of the brain. He had no clue what it was. And he never actually said, because he discovered the disease, he never said, oh, I'll call it Alzheimer's disease. Okay. Nowadays, scientists always put their own name to whatever they invent, right? Fine, yeah? But he never did that, okay? So many years later, when people actually realized what he saw, they gave his name to the disease. What happened, so when he was presenting this data in 1906, something happened to him, and I hope you won't let me suffer the same that what happened to him at the end of the talk. So he gave a presentation about Auguste Deter and about what he saw, and there was not one single question afterwards. So if there is no question after a presentation, that can only mean two things. Either the presentation is really bad, or nobody cared what he said, right? And nobody cared about a 50-year-old woman who got weird. Okay, who cares? In the early 1900s. So nobody was really interested in his findings, and that was a problem for him. Because if nobody's interested in your science, you will not get funded. If you have no money, okay, that was it with your science. But Alzheimer, as you all saw that, was a very attractive, handsome man, and he was married to a very rich lady, and she wanted his science. So that was but that was the first publication about the disease, and which Alzheimer described. So these are this is the lunatic asylum where Auguste Deter was housed. These are the original bathrooms. These are the gardens, and these were the brains the doctors collected after the patients died. Alzheimer's disease is a disease that affects pretty much everyone. And here you see some people who suffered from the disease and now are all dead and died for various reasons, but they all had Alzheimer's disease. Margaret Thatcher, John Heston, Helmut Schön. He was a famous German soccer coach and a football coach when we won in '74 the championship, Rita Hayworth and Ronald Reagan. From which person did you know that he had Alzheimer's disease? Was she? Was that no one? Anyone? Margaret Thatcher, right? So all the other ones here, and even with Thatcher, it was not really common knowledge. And that is really a problem. You have very famous people, and you have no clue that they, or in general, people have no clue that they suffered from this disease. And for the longest time, what happened was when people suffer from Alzheimer's disease, they get weird in your eyes and our eyes. But in the eyes of the family, it's a problem. It's a problem in the way, and for yourself it's a problem. You realize there's something wrong with me, and you start to hide. The family is starting to hide you from everyone else. And then you're suddenly gone. Like, for example, Rita Hayworth. If you don't know her, in the 50s and 40s of the last century, she was a Hollywood icon, maybe like Angelina Jolie today. And she was gone like that from one day to another. Nobody has seen her ever again. And she suffered very early on from Alzheimer's disease, but she was so ashamed, and her family was so ashamed, that she actually were hiding herself from the public. And that happened to most of them. And to him, I have to say a word, because he's a problematic figure. He was the American president when I was young. And I was protesting in the streets against him. And then I went to the US. Many years later, I worked lived in San Diego for almost 5 years. And there I realized the Americans have a completely different view to him as we have. And it's very interesting, actually, because he was diagnosed with Alzheimer's disease when he was an active president in the Oval Office. So when he was American president, he was diagnosed with Alzheimer's disease. And then think about it. You're an American president or you're a Russian president and you have Alzheimer's disease. What would you do? You would go immediately to all the well-known physicians in your country or worldwide. Because, I mean, you are so powerful, we cannot even imagine. So there is really no option, no obstacle for us. We could get any treatment we would ask for. And that's what he did. And then he realized there is no treatment. And then he asked himself, am I the only one who has Alzheimer's disease? No, millions of people have Alzheimer's disease. So why does nobody know about that in the 1980s, right? And then he said, okay, there's something clearly wrong. And he said, he made it public. He said, okay, I'm your president. After he resigned, immediately he came out and said, okay, I have Alzheimer's disease. And what you can do is you can watch me suffering from that disease. So he was not hiding, he was basically going out and was doing everything an Alzheimer's disease person would do in public. And he was not ashamed of it, his family was not ashamed of it, and because of it, the public, the journalists got aware of the disease, and a lot of money was going into Alzheimer's disease afterwards, right? But it needs people like that who come out and say, okay, here is a problem, we have to do something about it. And that's what he did. So there are always two coins of a medal. I had to realize when I grew older. I still don't like his politics, but I liked what he did for the disease. Similar person. Charlton Heston, when he was young, he was walking with Martin Luther King and fighting for the freedom for the African Americans in America. When he was old, he was the president of the National Rifle Association, and he claimed that every American should wear a weapon and should be able to shoot anyone at any time. Okay. But interesting. Has anyone maybe seen the movie Bowling for Columbine. It's in a movie. It's an American movie about a massacre at an American school, and the director made a huge mistake in that movie. The movie is excellent, but they interviewed Charlton Heston in that movie, and he was the president of the National Rifle Association, and was always telling everyone, okay, wear guns, shoot back, whatever. But at that point, he was severely affected with Alzheimer's disease, and you cannot interview a sick person. Whatever stupid thing they say, it doesn't matter. They were sick, so never touch anyone or who is that sick and make fun of it. So that was a problem. But here, I mean, all these people have Alzheimer's disease. Can you realize one thing that they have in common?
People,
only people? Just people? Yeah?
Famous?
Famous? Famous? That would be cool if only famous people get Alzheimer's. Then we would be all protected. No?
They're white. They're white.
They're white. Oh, good point. Oh, next time I'll put another... They're old. Right? They're old. So aging is the biggest risk to develop Alzheimer's disease, regardless which education you have, regardless where you're born, regardless who your parents are. The color of your skin doesn't matter. Right? Everyone gets Alzheimer's disease. You just have to get old enough. And so this is just a diagram of the worldwide oldest people from different countries, and at the moment we are somewhere here, and I think, which color is that? Icelanders might be the oldest people. No, Japan. Actually Japan, right. In Japan are the oldest people these days. At the moment, there are more than 50,000 people in Japan above 100 years of age. Why they get so old, we have no clue. But in Japan, they are the oldest people at the moment. So if aging is a problem, then we look at the aging in the whole world, and we project that for 2050, right? The darker the color, the older the population. Meaning, in Russia here, you will have 104 million people, approximately, in 2050, and 38 or 40 million of them will be above 60 years of age. Meaning, 1/3 of the population in Russia in 2050 will be close to retirement. So that will be, of course, a problem for any kind of retirement fund. Nobody will tell you that, but it's pretty clear. And it is a problem for any kind of disease that is associated with aging. Cancer, all neurodegenerative diseases, everything goes through the roof when you're old. So we have a real problem in hand here. If you look at Japan, for example, there will be 109 million people, but almost 50 million of them will be above 50. So half of the population of Japan will be almost close to retiring in 2050. So that will be a problem, can be sure. Germany is the same. So this is a retiring map of Germany. And the thing that will happen in Germany is we will have, the darker the color, the older the population, and especially here in the eastern part of Germany, we have a lot of people in 30, who will retire. That is pretty obvious from the color. So why is that the case? That was the case after the unification of Germany. Most of the young folk went to West Germany, and the older ones stayed in East Germany. And now we have that problem that we will have in the near future a huge part of Germany that will be completely living on retirement funds. So this is not a medical problem, that is a societal problem. And we have to think of all these issues that might have, that will certainly happen in Germany. I'm not so knowledgeable about Russia, but I'm sure it might be happening here too. So I looked up some numbers for Russia. This year is the population development for up to 2011 in Russia. And you see you have a lot of people in Russia at the moment, those days, which are around 50 or end of 50. And only little people were born. So in the near future, you will pretty much have the same problem. Except I found this curve here, and that means this is the average number of people, meaning you have as many deaths as newborns, right? Then you keep this average line. And it was dropping down dramatically, but now it's coming back up, and you actually have more newborns than people die in Russia. So that is a good development here in Russia. And in Germany, we are far from that. It's really tough. If this is a problem, if aging is a problem, does the community, does the society know about it? Yeah, this is in German again, but the biggest fear of Germans are, number one, poverty among the elderly. Okay, there already is aging in there. The second number is natural disaster. That I don't understand. I mean, we have some floodings from once in a while, but we have no earthquakes or anything. But okay, natural disaster, second. Third, in need of care and dementia. So there we are already with Alzheimer's disease, right? The third biggest fear of the German population. So if that is such a big problem, does the government do anything about it? In Germany, in Russia, in the United States, wherever. I'm pretty sure you can pick the numbers from any country. These are the numbers from the United States. In gray here, this is the amount of money the country has to spend for the care of Alzheimer's disease patients. This here is the amount of money they put into research for Alzheimer's disease, right? And this is here a little bit bigger, so that you actually can see that. You have more people affected by Alzheimer's disease every year than cancer, AIDS, and heart disease together. This is the amount of people who have problems with Alzheimer's disease, newly developed Alzheimer's disease every year. So there's a huge number of patients, but nobody cares, right? And the problem here is, why are we putting so much more money into all the other diseases? Like, if you see here again, Alzheimer's disease, research amount, this is the amount into hospital care, right? Cancer, pretty even. We're putting a lot of money into cancer research, which is good, don't get me wrong. It's fantastic that we put a lot of money into cancer research. I mean, there's no other disease, to my knowledge at least, where we made so much progress in the last 10 years. There are so many cancer types that are curable, and we're making progress in that term. So that is fantastic. Heart disease, we could do more. AIDS, even the opposite. We're putting much more money into research for AIDS than the patients actually cost the society. So why is that the case? Well, when you're old, you don't count anymore for society, right? You get Alzheimer's disease when you're old. When you're 70, when you're 80, you don't produce anything anymore, so why should we care? Right? That's terrible. But there is no lobby for old people. When you get cancer, you're most likely still productive. You're young. You have a lobby. AIDS, you're certainly young, right? So that is a problem. Now, so we have to convince the governments to put much more money into research for Alzheimer's disease. But one other aspect I wanted to talk to you about is how do we detect whether somebody develops Alzheimer's disease? So there are some clinical tests that we can do, and you can Google them, it's pretty easy to find them. But you can do other things. So maybe your aunt, your uncle, or your grandparents come to you and say, oh, I lost my keys, I'm sure I have Alzheimer's disease. And say, no, I have Alzheimer's disease. But you can test that. You can test that pretty easy. So a common test for Alzheimer's disease is you ask a person to draw, for example, 11, 10 on a regular, and they should draw a clock and put in 11, 10, right? And if they can do that, no problem whatsoever. No Alzheimer's disease patient can do that. I can show you that. This is the drawing of the same person 1 year later. So at that point she was diagnosed normal. 1 year later, severe Alzheimer's disease. With Alzheimer's, 1 year earlier, this was drawn for her, and she should redraw this cubicle, and she could do that easily. 1 year later, no way that she could do that. So our diagnostic tools are really terrible. So we can diagnose Alzheimer's disease. Well, let's say 5 years ago, we could diagnose Alzheimer's disease when anyone could do it. Right? You didn't need a doctor for it. We could see that person had Alzheimer's disease. These days, we can diagnose it maybe a year before anyone else can do it. But that's still too late. But how can we do that a year earlier? So this person here, she was a lady from Mainz, from my university, and she started with this, she came in for a routine check, and what they did as well with her was they did a PET scan. A PET scan is a scan of your brain, and your brain, I told you, uses a lot of energy. And energy in the brain is basically, the source for energy is glucose. So what you do in a PET scan is you apply to the patient radio-labeled glucose. And you can image then the brain and see where you have certain counts, decays from the radioactivity. And then you can quantify active areas of a healthy person or inactive areas of a sick person. And a PET scan, an original PET scan, is here. Good, there. So this is what comes out of a scanner. You have no clue what that is. It's just colorful, right? And every doctor has the same problem. They can't see anything on it. So what you have to do is, or what people do is, they scan hundreds and hundreds and hundreds of healthy people and put the scan from all these healthy people as a threshold in black. Then you just take a scan from anyone, put it over the threshold, and everything that lightens up is inactive. Right? So this is an inactive area of an Alzheimer's disease patient. And at this time here, she was still capable of drawing that. Right? But in the PET scan, we could see there is a difference in the brain. 1 year later, when she was drawing that, the area only got a little bit bigger. Not much, right? But it was enough to convert her from a normal to a severe Alzheimer's disease person. So a PET scan would give us the possibility to detect Alzheimer's disease earlier. But think about it. You cannot inject into the whole population radio-labeled glucose and then image everyone all the time. And that's not an option, right? So we have to get better, and even much better than that, what we have to know is whether you develop Alzheimer's disease with 55 and you with 85. And we have to know that now, right? If we think about it with 55, you have already Alzheimer's disease. So we need an early marker that we can clearly distinguish who will get Alzheimer's when. If I would get a marker and tell you get Alzheimer's disease 95, you say, well, I don't care. I'm dead anyway then, right? But if you have a clear marker which tells you certainly you will get Alzheimer's with 45, okay, 45 is a different option, different problem, I'll show you later. But 55, then you would care, right? Or 65, you would care. But we don't have that. We have no clue. And that is the biggest problem that we face at the moment. Okay, so the whole series here was called Molecular Aspects or Fundamentals of Alzheimer's. So we haven't said anything about molecular yet. We have to start with that now. So we go back to what Albus Alzheimer saw in his images. And what he saw was basically these plaques and these tangles. And we start with these tangles here. Neurofibrillar tangles are structures that are coming from microtubules. Microtubules, you know, you have in your axons to transport kinesin or dynein vesicles along the axon. And they're usually stabilized by a small protein, which is called tau. When you phosphorylate tau, you put a charge on tau, and with that charge, tau gets detached or gets loose from the microtubules, and the microtubules break down. And when they break down, they build these neurofilament, these tangles here, which you see there. And that is one aspect of Alzheimer's disease, that your neurons break down, and you have a neuronal loss and a synapse loss in Alzheimer's disease. So that is one key aspect of Alzheimer's disease. Okay, so the next we have to discuss is this here, the plaque. But since I checked, luckily, my watch, otherwise I would talk forever, I would suggest we talk about that after the break. Is that okay, Anya?
Sure.
Okay, so we have a break now, and then afterwards we will go into more details of plaque formation and later on into therapeutic approaches. So we stopped with the tau protein, and the tau protein is very, very, very important since these little tau proteins act as a clamp and keep the microtubules together. But this is as much as I will tell you about tau, because I don't know much more, to be honest. Tau is an important protein in Alzheimer's disease, and a lot of researchers are working on it. And if you find any kind of cure which tackles tau, fantastic. But I can't do it, I don't know anything about tau, right? So now we are getting even deeper into molecular aspects of one part of the disease that I know of. And that is an aspect that is related to genetic modifications. So there are certain genetic modifications of some genes in your body that will ultimately leave you with Alzheimer's disease. No questions asked. So these are familiar early onset genes, meaning if you have a mutation, a certain mutation in one of these three genes, you will develop Alzheimer's disease. So you have the APP gene. Mutations in that gene will cause Alzheimer's disease as early as 35 to 40 years. The presenilin gene. There's one patient with a mutation in presenilin 1, and she developed Alzheimer's with 17 years of age. So that is a big problem. The question really is, why do get people Alzheimer's disease if they have a mutation in one gene? When you look at this gene here, APP is the name of the gene, and it's localized on chromosome 21. Most of you know a disease that is associated with chromosome 21, trisomy 21. We'll look at these patients in a minute. So what is APP? APP stands for amyloid precursor protein. And it is a protein that is called a type 1 transmembrane protein, meaning it's a protein that reaches through the plasma membrane, and the N-terminus of the protein is outside the cell, and the C-terminus is inside the cell. And this protein is generated in your body in every single cell since you're born. And we don't know why. We have no clue what the physiological function of this protein is. So we know it is conserved in C. elegans, Drosophila, hamster, mouse, elephant, whatever, human, primates. There's a very high homology in that gene. But we don't know the physiological function. But if that is conserved during evolution, we can be pretty certain that it is not carried through evolution that a worm develops Alzheimer's disease. So that can't be the reason to carry that gene. So there has to be a physiological function, but we have so far no real clue what it is. This gene encodes for proteins of different length, and the number here just represents the amino acids, the number of amino acids within that gene. So under normal conditions, this gene is processed, or this gene encodes for protein, and this protein then is processed. Processed in this term means cleaved by enzymes. And 99% of the time in your body, all the time, this protein is cleaved by an enzyme, which is called alpha secretase. And when that enzyme cleaves, you have a small fragment that is stuck in the cell, and a large fragment that just floats around your body. Why? We have no clue. This happens all the time. And if that, unfortunately not all the time, if it would happen all the time, you don't have Alzheimer's disease. Because 1%, or maybe less percent even, there are two different enzymes that cleave, and these are called beta secretase and gamma secretase. And when these enzymes cleave, this APP, this amyloid precursor protein, this fragment here will be released. It's the A-beta peptide. And the A-beta peptide accumulates and forms at the end these plaques. And these plaques were one of the hallmarks of Alzheimer's disease. These days we know that the plaque himself is not really dangerous. What's dangerous is the soluble parts of these proteins. So if you have a lot of these proteins, they stick together, form dimers, trimers, oligomers, and as long as they are soluble, you have a problem in terms of that you degenerate your neurons. Why? We don't know. So there is no receptor known that triggers a cell death signal to a neuron. We don't exactly know what's going on. But we know if you have a high amount of soluble A-beta peptides, you have a higher risk to develop Alzheimer's disease. And any mutation within the APP gene, which I said was on chromosome 21, will lead to early onset Alzheimer's disease, so with 35. So what happens there? For example, here in brownish, you see the amino acids that encode for the A-beta sequence. If you have a mutation right before the first amino acid here, from KM to NL, which was found in a Swedish family, therefore it's called Swedish mutation, you have a higher chance that beta secretase cleaves at this position. If you have a mutation back here, which is called the London mutation, it was found in a family in London, you have a higher chance that the gamma secretase cleaves at the end here. So you generate, with every mutation here in the APP gene, you generate, theoretically, more A-beta peptides. When you generate more A-beta peptides, you get Alzheimer's disease early. So that is the first proof, to my mind, that a mutation that causes more A-beta peptides causes Alzheimer's disease. Right? Therefore, I would say this is pretty good indication that a mutation in this protein that either gives us a higher affinity to beta secretase or gamma secretase is one reason that Alzheimer's disease actually develops. So that is only one gene we were talking about. So what happens here? Let's try to sum that up, what I was talking about. We had in the first aspect alpha secretase that cleaves. We have this soluble part, and we have a C-terminal stop that is still in the membrane that can later be cleaved, and some other aspects of these proteins will be released. But this is non-toxic and therefore non-amyloidogenic. The problem starts here when we have beta secretase. For example, we have a mutation in APP. This enzyme cleaves more often. Therefore, we have more C99 stop, and gamma secretase can cleave with more A-beta peptides, right? Or we have a mutation down here, and then gamma secretase can cleave more often, or cleaves very unspecific. Gamma secretase, unfortunately, is an enzyme that doesn't cleave always exactly at the same position, but cleaves at different positions. And when it cleaves even a little bit further down here, you don't have an A-beta peptide of 40 amino acids, but you have an A-beta peptide of 42 amino acids. And 42 amino acids, that is a toxic peptide. If you take this peptide and add that to cells, your cells die. So that stuff is pretty toxic, and you find that in the brains of Alzheimer's disease patients. These 42 peptides. So let's do a little biochemistry. If we say we want to test for these fragments in cells, in tissue culture, or in neurons, or in the brain, you can grind up the brain and then do a SDS page and the Western blot afterwards. So what we want to test first is alpha secretase activity on APP processing. What we do is we run a gel, then we take an antibody, and for example, the antibody would detect down here. Then we would clearly see these C-terminal fragments. But when the antibody detects down here, it would also see APP full length. That's what we see up here, right? So that is APP on a biochemistry level shown in a cell. Now, we use a different antibody, which detects, for example, here, right, in that position. Then we would see the soluble part of APP. APP is alpha. And we would actually see as well down here a beta peptide, because when the gamma secretase cleaves, beta and gamma secretase cleave, we would generate a beta peptides, which we can see here, even with the same antibody, right? So it's, for us, really easy to detect these peptides, but we can still not say when anyone will suffer from this disease. So in tissue culture, piece of cake. Huh? Yep.
In any tissue or some special tissue?
In any tissue. So that is a problem. So think about it. You have this protein really everywhere. You have these cleavages everywhere, but you have never heard of plaques in your heart or your liver, right? We don't know why. What we think is going on is, as soon as these A-beta peptides reach your bloodstream, they will go to the liver, taken up by the liver, grind it up, and it's not a problem at all. But in the brain, that's not happening. The brain has no direct access to the blood. You have the blood-brain barrier, and we will talk about the blood-brain barrier one of these days. So the beta peptides cannot just go across the blood-brain barrier and can get cleared from the brain that easily than from any other organ. So that might be one reason why we develop Alzheimer's disease in the brain, that the beta peptide can't get out of the brain, but from any other organ it goes into the blood and we have no problem whatsoever. Right? And that might be the reason why we develop Alzheimer's disease when we're old. So over time, you accumulate more and more and more A-beta in the brain. For when you're young, your blood vessels are healthy and okay, and you transport some A-beta out of the brain. The older you get, your blood vessels go down, let's say that. And you know that from all kinds of heart diseases. right? And if that happens, your risk to develop Alzheimer's disease increases due to the fact that you probably have more beta in the group. So one other aspect that I believe that beta is the driving force in the disease comes from trisomy 21. Trisomy 21 basically for Alzheimer's disease means every patient with trisomy 21 has 3 copies of the APP gene. Most of the trisomy 21 patients will develop Alzheimer's disease at the age of 40.
So if you ain't got a trisomy, you ain't got no Alzheimer's disease?
No, you will get Alzheimer's disease with 40. So most likely, due to the fact you have 1 additional copy of the gene, you produce more APP, you have the same amount of enzymes, but you have more substrate for the enzymes. So you produce all the time more and more and more A-beta, you will develop Alzheimer's. Yeah?
Excuse me, do you mean that every patient with Down syndrome will have this disease?
Yeah, Down syndrome, trisomy 21, same name. So they all will develop Alzheimer's much earlier due to the fact that they have more A-beta. Only because they have 1 more copy of A-beta. So another reason why I believe A-beta is the driving force of the disease. Right? So we have genetic mutations, more A-beta, early onset Alzheimer's disease. Trisomy 21, additional copy of APP, more A-beta, early onset Alzheimer's disease. The reason why I don't talk about tau much more is because I think tau is very important, don't get me wrong, but A-beta is the first driving force in Alzheimer's disease. And we can show that in animal models, that when you have a lot of A-beta, you will get phosphorylated tau, and then that all together is toxic. If you have an animal model where you start out with phosphorylated tau, you will never get an A-beta phenotype. You will never see plaques or anything like that, right? So that's the reason why we think, at the moment, A-beta comes first, then tau, and then toxicity. So therefore my interest always is, at the moment, to get at the root of the generation or the clearance of the A-beta peptide. So, I wanted to say something else. Well, it doesn't matter. Maybe it's a little later. So switch gears from the familiar early onset genes to these genes down here, late onset genes. So, meaning, if you have mutations in these genes, you will not automatically get Alzheimer's disease. But it's a pretty good marker for Alzheimer's disease. Especially, and the best marker we have at the moment is this one, ApoE4. ApoE, some of you might know that protein, is involved in lipid metabolism. So it is involved in cholesterol metabolism, if you wish. You have ApoE in different isoforms. You have ApoE in the isoform ApoE3, 2, 3, and 4. And the only difference is 1 amino acid. So one exchange in the nucleotide composition will give you different amino acid exchanges in the whole protein. And this exchange will have tremendous problems for you in terms of Alzheimer's disease. Because if you are ApoE homozygous, so 2 alleles of ApoE, your risk to develop Alzheimer's disease will increase by 70%. We don't know why. But this is a huge problem. And knowing that this is the best known genetic marker for Alzheimer's disease, the German government was actually thinking of a mandatory test in the whole population, genetic screen for ApoE4. What do you think of that? Let's say in Russia there would be a mandatory genetic test for ApoE4. Would you like that?
can we do after that?
That's my question to you. So you know that you're APOE4, and then?
So you can put
Nothing.
more money in research.
Only if you're rich.
You can do PET every year.
Every year.
And spend your money.
Okay, so you think it's useless?
I don't know. There are no prophylactic things.
You can learn to accept over time.
So you would think, if I understand you correct, if you are APOE4 positive, you would think you will get Alzheimer's disease. Probably?
70%.
70%. So does it mean you get Alzheimer's disease?
No.
No. Right? So they give you a risk to develop Alzheimer's disease, but that doesn't mean that you get Alzheimer's disease. Right? You live in fear that you will develop a disease where we have no treatment. What would that be good for?
Better not to know.
Exactly, better not to know. My mother, she did that screening, and afterwards she called me by phone and told me. I said, I don't want to know. It was too late. So I know 50% of my genes. If I have a treatment, fine. But if I have no treatment, think about it. You do that screening, and you have a health insurance, and you are APOE positive, APOE4 homozygous. I'm pretty sure any kind of health insurance will kick you out. Why would they insure an Alzheimer's disease person? So all these are real risks in society, and luckily the German government never really did it, so we can all live in peace. But I mean, if you think of all of these consequences, it's really terrible, right? So APOE4 is the biggest risk factor, genetic risk factor that we know of. You can test, of course, you can test yourself. It's not a big deal, I think. A whole genome test is about $100 to $150. You can send your blood sample and they will screen your whole genome and then you know. Yeah.
Why APOE4 is more sensitive? Because of frequency in population or what?
No, it's actually quite rare, right?
But in general, in Alzheimer's disease.
Yeah, that's a good question. I don't know.
Okay. No, I just don't understand. You can have alpha-2 microglobulin, APP, and also APOE4.
So this is no. This here is the best known risk factor, right? These are others which are more or less validated. This one here is 100% sure. If you are APOE homozygous APOE4, your risk is 70%.
It mostly connects with the disease.
Yeah.
It's like specific.
Yeah. No, it's not specific. 70% is not specific,
Yes. Okay.
right? So this is just a risk factor. Why? We don't know.
And is APP specific?
That mutation?
Yes.
Yes. So the genes up here, if you have your mutations there, your risk is 100%. It's actually not a risk. You will develop the disease, right? So that here is a mutation, you will get the disease. But don't get me wrong. All of you have that gene, and all of you have that protein in every cell. Hopefully not mutated, right? So since you produce A beta all the time, at some point in time, you have enough A beta. When you're old, you develop the disease. Okay, here, this gene, next problem. You develop Alzheimer's disease with 17 years of age. So what is presenilin 1? Presenilin 1 is a protein that looks like this, and it is really the enzyme that is the catalytic domain of gamma-secretase. So this is here the catalytic cleft where APP is inserted and cleaved. So now you have a mutation in an enzyme that produces more A beta again. You will develop Alzheimer's disease with 70. So another good explanation why A beta is a driving force in the disease, right? We have mutation in APP. gives you Alzheimer's disease. Mutation in an enzyme that cleaves APP gives you Alzheimer's disease. An additional copy of APP will give you Alzheimer's disease. So, for me, that is pretty solid, right? The reason why I'm always saying that is because there are a lot of people who don't believe in that. Okay, there are a lot of people who say APP has nothing to do with the disease. I don't agree with them, but I very much appreciate that people say that. Because if we all run into the wrong direction, we really don't have to hurry, right? And there always have to be people who say, you're wrong, I'm going into a different direction. That's science, guys, right? If we all agree, that we won't, there is no progress. We have to disagree, and we have to learn to disagree.
So what are the reasons to think differently?
The reason is they agree with that APP is the driving force of familiar Alzheimer's disease, so when you get Alzheimer's with 30 or 17, but they disagree when you're 75 or 80, because there is no genetic proof for an 80-year-old to develop Alzheimer's disease due to that mutation. So Alzheimer's disease, they say, could then happen through all different kinds of reasons, right? Which is possible. I have no proof for that. But since the histopathological hallmarks between a 17-year-old and an 85-year-old are identical, and the biochemistry is identical, I would say it's the same cause, right? But only because I say that doesn't mean that's true, right? So presenilin is the protease of this huge complex. So gamma-secretase exists out of 4 proteins which cleaves different proteins. And that is a problem. Because gamma-secretase cleaves APP, but cleaves up to 50 other substrates. So why is that a problem? That is a problem because gamma-secretase cleaves, as I said, APP, but cleaves as well Notch. Notch is a protein you probably have never heard of.
Migraine? Migraine?
No?
Notch.
Notch is
What is that?
involved in developmental aspects. Cell migration, proliferation, differentiation, right? And if you have a disturbance in Notch processing, you will develop cancer, for example. So, gamma-secretase cleaves Notch in exactly the same way as APP and many other proteins. So it's a very promiscuous enzyme and not specific at all, which is a problem in terms of therapy, which we will talk in a minute about. So what do we know about Alzheimer's disease now so far? This here is the so-called Alzheimer's disease or amyloid cascade. What we believe at the moment is APP is the starting point, and from APP we generate A-beta peptides, and the toxic ones are the A-beta 42 peptides. And these peptides are either deposited in forms of plaques, which is most likely not a problem, or they are formed in so-called oligomers. Dimers, trimers, oligomers, and they're toxic. And they then, this will result in some kind of cellular stress. You will get paired helical filaments. These are the tau filaments I talked about. And from there you get neuronal dysfunction and death, resulting in dementia. Right? This is the so-called amyloid cascade. We have no definite proof that that is the case, but at least many people believe that this is actually the process how Alzheimer's disease develops. This here is a graph on what we know at the moment. So when we think of Alzheimer's disease, we always think of clinical diagnosis, and clinical diagnosis is shown here in And when you look at other aspects here, here's clinical diagnosis, and we have amyloid burden. Amyloid burden means the amount of A-beta peptides. You have already a lot of A-beta peptides in your brain when we can diagnose Alzheimer's disease. NTF, neurofibular tangles, so means tau. You have a lot of tau already in your brain when we diagnose Alzheimer's disease. And here, this line, even scarier, neuronal and synapse loss. You have a lot of synapses and neurons already lost when we diagnose Alzheimer's disease. So that is scary. You lose even 20 to 30% more neurons that you would usually lose during aging in Alzheimer's disease. We have to detect Alzheimer's disease here, right? Before we have that increase in A-beta peptides. Yeah?
Excuse me, this is for a clinical test, for example, clock test. Yeah?
Mm-hmm. If we go now with a PET scan, right? We are maybe there. Okay? We need, this year, the increase in amyloid peptides is at least 15 years before you develop the disease. Right? So this is 15 years before. So this, we need probably to go back 20 to 25 years before you develop the disease to have a clear marker to tell me you were 55, right? Alzheimer's disease, and you're 95, if I remember correctly. We have to know that early. Then we have a chance to tackle the disease. But we have no clue yet, right? We are here, which is really nothing. So if we have any clinicians here, or medical doctors in SPE, what we need from you is a marker, guys. I need a marker who gets Alzheimer's disease early. Otherwise I can't do any progress. Yeah.
And what about antibodies tests? You were talking about.
Yeah. So what you can do is you can puncture the spinal fluid, right? And then you can detect the amount of A-beta. You can do that.
Yeah. So where you will be doing this, like on this?
Well, you can do that anytime. We could do that now with you.
Yeah. So yeah.
But then we are here,
So,
right?
yeah.
So then we have to do that every year.
What's the point?
To everyone.
How you can provide?
So do you want to really puncture your spinal fluid every year?
No.
I mean, it's possible. The problem there is infection, right? If you carry any kind of infection, you have dramatic problems. I wouldn't do that. That is not a perfect way for a clinical marker. But I agree with antibodies. What we should need, for example, blood sample, right? If we could get anything out of a blood sample, fantastic. But so far, all blood markers don't work.
So on the spinal.
Yeah, more or less. More or less. So nobody's doing it. So, now we try to think about therapy, right? This is, the green line here is regular brain aging. The older you get, the more your general cognition declines. That's normal. And here we have normal aging, and then at some point in time, Alzheimer's starts. We have no clue when that is. Here we can detect something, because then we have MCI. MCI means mild cognitive impairment. Mild cognitive impairment means something is wrong. Out of mild cognitive impairment, all different kinds of diseases can develop. Parkinson's disease, Huntington's disease, whatever, right? Alzheimer's. Doesn't mean that you have Alzheimer's disease. So at some point in time, we can detect Alzheimer's disease. Far too late. And from here to here is about 7 to 9 years. Meaning, when we these days detect Alzheimer's disease, most of your decline, of your cognitive decline, you will actually realize as a person, as a patient. Only in the very last years, you maybe have no clue what's going on. I'm saying maybe because we can't talk to these people anymore. We don't know what they really realize. But for the longest time, these people suffer. And suffering means they lose everything. They lose their personality, they lose any kind of reflex except sucking reflex and grip reflex. These are the reflexes of a newborn child. Every other reflex is gone. So Alzheimer's disease patients in the severe state, when they cough, they actually forget to breathe and they die, right? Which is normally a reflex. So the brain completely deteriorates, and for the longest time you realize what's going on. That is really terrible, guys. So what can we do? Since we can at the moment only diagnose Alzheimer's here. Ideal would be full cure, right? I would say, you can prove me wrong again, but I would say absolutely impossible. Because at this point in time, you have lost an additional 20% of your neurons. They will never grow back. I mean, okay, then people will prove me wrong again, but if you listen to the news, right? Everyone, Dina, I'm sorry to say that, everyone is working on stem cells, right? And stem cells will cure every disease. If you work on stem cells, you hear on the radio, it's good against Alzheimer's disease. How? Would you really think you have a deteriorated brain, then you inject some stem cells and new neurons will form that give you your memory back? No way. At this stage, right? At this stage. We're talking always about this stage. So I'm very, very, very suspicious when people come up with new, fantastic possibilities how to treat a disease based on stem cells. To work with stem cells is fantastic, but to sell everything that it will cure Alzheimer's disease, I don't know. Can we get symptomatic benefits? Yes, we can. There are some drugs that give some patients some relief. A lot of sums in there, but it's really it's a help for short term. What might be possible, which would already help a lot, is if we could slow down progression of the disease. So think about it. We would develop a molecule that would inhibit a beta peptide generation when you are old, right? When we detect Alzheimer's disease in your brain, then one can hope that no more neurons will die over time. So therefore, you decline slower. When you develop Alzheimer's with 85, and 7 years later, you're completely deteriorated, let's say it was 92, and we could slow down progression, you might be fully deteriorated then with 100 years. That would be good, right? Because usually who will get 100 years of age, except some Japanese? I mean, that would already help a lot if we would give the people another 5 years, let's say that. That would help tremendously at the moment. Whether that's possible, it's a whole different issue. So let's talk 15 minutes about therapy. What we do at the moment. So these are different aspects where we could go into, try to do therapeutics. The first thing that all the pharmaceutical companies are doing are inhibitors for enzymes. It's easy for them, they generate them. So gamma-secretase and beta-secretase inhibitors you can generate easily and apply to patients and see what's going on. And that's done at the moment, and there are huge trials going on to see whether that works. And I'll tell you a little bit about that in a minute. One can try to use chelators. Chelators are molecules that capture cations. So zinc, copper, for example, because if you have cations, you have a faster aggregation of the beta peptides and therefore probably higher toxicity. I'm not so sure whether that's a good way to go, but anyway, people try that. One can try to increase clearance, so to get more A-beta out of the brain, or to do a vaccination against Alzheimer's disease. I'll show you some results of that in a minute. For the longest time, people have tried to tackle the oxidative stress that accumulates in the brain after you accumulate A-beta peptides. So antioxidants, vitamins, for example, some food restrictions were thought to help during Alzheimer's disease. I'm not so sure whether that works. So I've never seen any clear-cut study that really shows that that vitamin really helps in the disease. So I would be very careful. And one other aspect I want to talk about is about molecules that you all know and use that might help during Alzheimer's disease. So wait a sec, we will go into the general question. The general problem is you have a lot of A-beta peptides, and what basically happens is mutations result in more A-beta peptides and A-beta 42 peptides. And only a slight increase in the A-beta 42 peptide will result in very severe Alzheimer's disease and early on Alzheimer's disease. So even if you have only an increase by 30 to 50% of A-beta peptides, only a minor increase, you will develop the disease 20 to 40 years earlier than a regular person, right? So what we have to do is then decrease the amount of A-beta 42, and we should be fine. Okay, so pharmaceutical companies try to tackle beta secretase. I don't know whether you have ever had a biochemistry class, but this is a shortcut. So this is an enzyme, and usually in an enzyme you have an active pore. And your substrate goes in, and then your enzyme cleaves your protein, right? So theoretically that's correct for beta secretase. But the active catalytic site of beta secretase is not this big, it's this big. It's a huge catalytic site. So now try to develop an inhibitor. You can do that. It's such an inhibitor. Well, molecular aspects. Right? So there you have a problem with oral uptake. You can eat that stuff, but it's a peptide inhibitor. Put a peptide inhibitor into your stomach. Your stomach will celebrate, right? But it will certainly never reach your target. Or, okay, then you say, well, I don't eat it, I inject it. Fine, you inject it into the bloodstream. It will certainly inhibit beta secretase all over your body, but not in your brain, since the inhibitor is not crossing the blood-brain barrier. So we need smaller inhibitors, and some of them are in development at the moment and are tested in patients, and we will see whether that works or not in the near future. Gamma-secretase inhibitor, whole different story, easy, piece of cake inhibitors. So when we use our tissue culture models, for example, you have hundreds of gamma-secretase inhibitors. You just add that to cells, and then you see here, you add an inhibitor here, there's no A-beta whatsoever. Fantastic. So the pharmaceutical companies 10 years ago started to apply that to patients. All trials with gamma-secretase inhibitors failed because gamma-secretase cleaves 50 substrates. As I said, notch. And in some trials, people had a notch phenotype, meaning they developed skin cancer. So that can't be a therapy for Alzheimer's disease, right? So gamma-secretase is a tricky issue if you completely inhibit the enzyme. But there might be a possibility. So this is what happens. Gamma-secretase, as I said, is very unspecific and generates different A-beta peptides, different lengths. This one here, as I said, is problematic since high amounts will result in Alzheimer's disease. But usually you have a lot of 40, a lot of 42, very little of A-beta 38. So what we need is a substrate or a substance that binds to gamma-secretase and changes the activity of the enzyme. It should change the activity in the way that gamma secretase more likely cleaves in the way that more A-beta 38 exists. A-beta 38 is not toxic whatsoever. And if that happens, you have less A-beta 42. And you should have less Alzheimer's disease. Are these modulators available? Yeah. So this is a story that developed when I was in San-Diego, and actually in the lab I was working. We had a new postdoc in the lab, and he got the task from our professor to use NSAIDs for Alzheimer's disease. NSAIDs, if you don't know, ibuprofen is an NSAID. So we all thought that is the most crazy topic you can work on, right? And what happened was he put ibuprofen on his cells and measured A-beta and showed me his results because I was the senior postdoc in the lab. I looked at the results and saw A-beta is still there. Forget it. Stupid. Doesn't work. He did it again. Showed me. I said, it doesn't work. Again, nah, forget it. So I went home, came back next morning. He was still sitting there, bright smile in his face, and told me, blank, you're stupid. That's correct. So what he saw was the following. He realized that the total amount of A-beta stays the same, but A-beta 42 drops dramatically down. That would mean people who take high amounts of NSAIDs should be protected from Alzheimer's disease, right? Do these people exist? Yeah. For example, here, the Rotterdam study proved him right. Rotterdam study means in the city of Rotterdam, there is one huge area which is completely under medical surveillance. So any prescription, anything that people take is known. So you can go back into the data bank and check whether people were taking over many, many years high doses of NSAIDs. And arthritis people, patients, they of course patients, they take high doses and they have an 80% reduced risk of developing Alzheimer's disease. So, wait a second. Don't go into the pharmacy and eat ibuprofen like hell now, right? Don't never ever do that. I mean, that's really dangerous. Don't do that. I'm just saying these molecules might work. So when this was published, Myriad Genetics became aware and licensed the patent for a drug that was called flurizan. Flurizan looks like ibuprofen. You see that, right? No, forget about it. So it looks like ibuprofen, but doesn't work like ibuprofen. Ibuprofen is an enzyme that is developed to tackle your COX enzymes in your lipid biochemistry. But this one doesn't do that. Doesn't tackle COX at all, but it tackles gamma secretase, as ibuprofen does. So this one should be actually possible to take over a long time, theoretically. So Myriad Genetics went into clinical trials, and they were so convinced that they had the cure for Alzheimer's disease. Actually, I was convinced too. 6-7 years ago, I told all the students, problem solved, we'll have something, right? And they were so convinced that they were really filming the opening of the trial results when all the CEOs and CSOs came together. They opened the results and it failed completely. And it failed because that one doesn't cross the one could have seen that earlier, but so there might be something that might work in modulating the gamma secretase. But there's a lot of work to do to convince other drug companies to do these trials again, because these trials cost hundreds of millions of dollars. And if one failed already, they usually don't touch it. So the last aspect we want to talk about is vaccination. Vaccination against Alzheimer's disease. When I heard that in the very first beginning, I thought, crazy, won't work. But then this paper came out in 1999. What they did is they injected into a mouse model with Alzheimer's disease antibodies. And these are the mice, usually when they develop these plaques, and you can measure behavior in animals, and they can't remember stuff, but when they were injected with these antibodies against the beta peptides, no plaques whatsoever, and the mice lived happily ever after. Meaning, theoretically, a vaccination could work. The first trials in humans failed completely due to really stupid mistakes we made, but nowadays, maybe there is an antibody or a vaccination that might work. 2 weeks ago, at the Alzheimer's disease, Parkinson's disease conference in Nice in France, there was a new substance introduced by Biogen, and it's at the moment in phase 1 trial, but it was a beta amyloid immunotherapy, so a vaccination, and what they showed for the first time in real patients, a tremendous reduction in A-beta, but they couldn't work with cognitive aspects, I think, but that looks really promising. And that looked that promising that 5 minutes after the presentation, the stock went up 10%. And that was pretty good. So maybe there's something coming. But again, it's very early. We don't know whether that is really working at the end. Okay, the last thing, clearance. We skip because we will talk about that anyway at the very end when we talk about the blood-brain barrier. And we stop now. And if you have further questions, don't hesitate to ask. Otherwise, tomorrow we will talk about more specific aspects. It will get more molecular, so it might be a little bit more difficult to understand. So I would be happy if you all come back, but I will understand if some of you will leave then because it's too hard. But I'll answer any kind of questions, so thank you very much for your attention. And if you don't want to ask in public, you can ask afterwards.
Yeah, good question. So, if you imagine there is a patient who have, for example, headache, and he take every week like an ACD.
Yeah.
So, is the risk of having Alzheimer's?
Yeah. So first of all, if you take aspirin, it doesn't.
Yeah, aspirin.
Yeah, aspirin doesn't. But the problem with NSAIDs is you have to take around 3,200 milligrams. In one ibuprofen capsule is around 200 milligrams, right? Over many years to get such an effect. If you do that, I'll bet your liver will tell you otherwise, right? So no, if you just for headache or if you take an ibuprofen every other day, that unfortunately won't happen.
So the patient will die from liver problems?
Yes, if you take now ibuprofen, you will die certainly earlier. It's another way of curing it, not getting it. Yep.
What you can say about memantine? Do you know this drug? It's only one drug that we use in what is common.
Honest?
Yes.
I don't understand how it should work.
Yes.
I mean, it's an NMDA.
Nootropic. Nootropic.
Yeah, so memantine is a drug that acts on the NMDA receptor. NMDA receptor is a receptor in neurons that is involved in memory building, if you want to say. And there is no causal reason why that should work, but that's not what they claim. They tell you just that you have a benefit for a short term of time.
But do they prove, like in a big trials?
Yeah, the trials all went through. They all worked.
So what should I suggest for patients? What kind of drug now? This year?
Okay, so the problem was the trials. It's not only memantine, you have the acetylcholine esterase inhibitors, right, as well. Those two are the only drugs that you can prescribe. They don't work, right? I mean, they may work for some patients for some time, but certainly not for all the patients. So what you have to do, what I would do, is if I could prescribe these drugs, I would prescribe them for a short while and watch the patient whether it helps or not. And most of the time, they don't help. So then, you don't prescribe them anymore. Right? If they help, keep going. In Germany, for example, the insurances don't cover them anymore because they don't work. But they're still prescribed by all the doctors worldwide. Since it's the only drug that was released against Alzheimer's disease. I'm really, really careful. It's a German company who makes them, but when you can eat ginkgo or anything else, might want anything.
But maybe I should prescribe ibuprofen.
No way! No way! You don't want to prescribe 3,200 milligrams of ibuprofen every day. No way! Right? I mean, you cannot do what you. No, you cannot do anything. But there was one question, I think it was you, who asked me whether it's correct that when you train your brain, you develop Alzheimer's disease later. That is correct. So I started in the very beginning to tell you that everyone will develop Alzheimer's disease, and regardless from your education, you will get Alzheimer's disease. Meaning, when you're educated and you're 65 and you're sitting in front of your TV, it doesn't matter, right? You just watch TV, your brain is not active. But if you're 65 and you start to learn Chinese or whatever, right, then your brain has really trouble to keep up, and it will work really hard. So a brain working hard means you have neurons that are connected through synapses. And the more your brain is working, the stronger your synapses will be. So now you're aging, you lose synapses, right? But you still have 3 other possibilities to get from here to there, so to get your information. Now you get Alzheimer's disease, you lose more and more synapses. But since some of them are really strong, you can actually get information for a longer time. Eventually you lose them too. Right? But if you're just lazy and watch whatever all the time, you lose synapses more early. And therefore, you develop Alzheimer's disease early. So that's the reason why we believe that mental exercise, cognitive exercise, later on, and you shouldn't basically watch Now TV all the time, and then with 65 stop and do something else and don't have it, right? But I mean, no, if you constantly train your brain, the risk to develop Alzheimer's disease decreases. It's no protection, but the risk decreases. Okay? Yeah?
You said something about metal halides. Are they useful and perspective or not?
Which one? Come again?
Are the metal halides?
Oh yeah. Metal chelators there was actually a study in Japan where they really did it in patients. What would you think would happen if you use chelators in a...
But it's everywhere, zinc and everything, so you can just chelate in the brain.
Yeah, so what happens? Nothing?
No.
Oh, there happened something, I'm pretty sure.
Some system polyorganic.
So metals.
Polymerase and all this stuff. Polymerase wouldn't work.
Polymerase wouldn't work. What else? Where do you have metals?
Yeah.
Breathing.
Hemoglobin and blood. Everywhere.
Everywhere. Magnesium, calcium, whatever, right?
Yeah.
People die.
Yeah.
So I wouldn't do that. But they seriously, they really tried that stuff. A lot of people died. So treatment for Alzheimer's disease, yes. They died early. Right? But no way, I wouldn't do that. Yeah.
What do you think about the association of diabetes mellitus and Alzheimer's disease?
Clear.
Does diabetes mellitus play a role in development of Alzheimer? Because I have read that ApoE4 plays a role in development of diabetes mellitus, and so maybe persons with ApoE4 and diabetes mellitus has an increased risk of development of Alzheimer.
Perfect. Yeah, it's correct. Everything you said is right. Why? I don't know. I would think with diabetes, this is now just really, I really don't know. This is, I would think it might have something to do with the vasculature, right? With your blood system, or with your endothelial cells. Because there you have in diabetes a problem. Otherwise, I have no explanation. So the insulin or sugar has nothing to do with the beta peptides, but that correlation is there clearly. But why, I don't know. Maybe you know more than me there in that perspective. I hope. Okay.
What's the phenotype of APP mouse mutant?
Is there a mouse mutant?
Is there? Should be. What's the problem? Is it
Mice do not develop Alzheimer's disease.
by default not, but there is another function. There should be some other function, so if it's everywhere.
Yeah, I agree. There should be some function. That was my PhD thesis actually. Physiological function of APP. I found one, but I don't know whether it's really correct. But anyway, no, mice don't develop Alzheimer's disease. And the difference between mouse APP and human APP, 2 amino acids. So theoretically, they should develop Alzheimer's disease, I would say, but they only live 2 years, right? So, and the regular biochemical processing is the same as in humans. So maybe they just don't live long enough. I don't know. But we only can induce a transgenic mouse model to have symptoms like Alzheimer's disease. So we put a human gene in there, overexpress it, produce more beta. But mouse mutants, I don't know why.
So there is no knockout for APP.
Oh yeah, of course.
I mean that.
Oh,
Yeah,
of course.
but mutants are knockouts.
Oh, okay,
conditions.
sorry, yeah.
Yeah.
So APP knockout has, in the very beginning, when it was first described, no phenotype. Now we know the mice have some small phenotypes. But the problem with APP is even bigger. We have 3 genes in the APP family. I didn't tell you the whole story. If you knock out 2 of the 3 genes, the mouse dies. If you knock out only 1, the 2 others take over. But a lethal phenotype doesn't tell you anything, right?
Does it die in the development already?
Yeah. So the knockout doesn't help us.
Conditional knockouts.
Conditional knockouts live, but there we don't see anything again. So
Even for 2.
yeah, and so it's APP is really a terrible protein, and it's, as I said, it's called amyloid precursor protein. But since we were all looking for the physiological function, everyone found a different physiological function. We termed it all-purpose protein because it can do anything, but we don't know what it is.
So, as I understand, we cannot just knock it down, because if we knock down this gene, the patient will die.
In a patient, okay, now we have a completely different picture. You want to knock down APP in a patient. Hands up, who wants their APP knocked down?
No, I don't want it. I just asked. what can happen?
I don't know. But knocking down APP in a patient would mean you have to knock it down in every cell, right? So when can you knock down a gene that affects every cell? Only when you start with a single cell, meaning with a blastocyst, right? We are not allowed to change genetic codes on a single cell basis because then we create life, right? We can't do that. And even if we would do that, we have no clue what will happen because, as I said, it is carried through evolution, certainly not for the fact that we will develop Alzheimer's disease. It has some physiological function. So I wouldn't do it in APP knockout at all in humans. Even if I would do it now. Yeah.
There is a theory that primary reason for development of Alzheimer's disease is a disturbance in calcium channels work. And what do you think about this? And how we can bind this theory to the forming called the A beta 42 and working of beta gamma secretase?
It's certainly clear that so calcium influx into neurons is completely altered in Alzheimer's disease. The problem is, as you say, we don't know how that works. So we know that the A-beta peptide somehow leads to neuronal loss, to synapse loss. But there is no receptor known, no real receptor known, that clearly shows us A-beta comes, binds, and then you have a disturbance in calcium channels, or you have a disturbance in microtubule organization, or anything. There's nothing known. It's only correlation. I can't tell you how the calcium is altered in that way. So it's really hand-waving that I'm doing here. I have no clue how it works. And nobody has. I mean, sure, some people would say they have, but I'm not really. Okay, I would say we stop here and continue tomorrow. Thanks a lot that you all came here. Excellent work.
Thank you. Thank you.